is from Kiyean Pharmaceuticals, Kiyean Nam. Welcome, Kiyean. Thank you.
Thank you very much for having me. This is my first CPTR meeting. It's exciting. Let me just start by introducing our company. You might not be familiar with. We are basically the venture company in Korea that happen to be working in TB area. One thing that we have 20 employees, fairly small company with the diverse set of a pipeline in different disease area. I think one thing that I want to emphasize here is that I don't see a lot of companies, a small company particularly, is working on this neglect disease area in TB area. And the small companies has incentive, financial incentive to work in this area, probably that the big pharma might not have one. So I just want to stress that it's better to have a more small company work in this area because and then the community has better to have some kind of exit strategy for this small company. We can work on this disease area that with the huge unmet needs. Okay, so Q23 looks like this, and then we have published this data in the 2013 for the first time. And if you look at the basic statistics, look at the basic data profile of the Q23, is that there's a fairly potent, is that CUM represent the outside of cell, and the KIM represent the inside the macrophage assay. And we see the very potent activity, 1 nanomolar, 0.5 nanogram, less than 0.5 nanogram per milliliter. And all the other basic physicochemical properties miss the drug likeness. And then we have been looking at the DDI potential from the beginning, and then there is so far no sign of DDI concerns in vitro level. And we also have tested compounds in the variety of the different the MDR and XDR strains. And so far that all the strains we have tested, it works. And we have an opportunity to test compounds in the TDR isolates found in Korea, 6 isolates, and it works pretty well in all these isolates. In addition to that, we are constantly screening the human isolate regardless of their drug resistance status. So far, in collaboration with the Korea Institute of Tuberculosis. And so far, we have tested more than 1,000 human isolates, and we so far have not found a resistant strain yet. Naturally occurring resistant strains, I should say. We tested the compounds in the anaerobic condition in 2 different settings, and then showing a fairly good activity in the anaerobic condition. Excuse me. And we also tested the compound against various different other microorganisms, and then none of the microorganisms actually respond to the Q23. That also includes the other mycobacterium, and then none of the mycobacterium so far we have tested is responding to the Q23. It's only TB and BCG. So this is one of the published data that we are testing in comparison to the bedaquiline, because that it was a part of the experiment that we did to find the mechanism of action of the compounds. So we tested various different things, and then happened to be that the ATP synthesis was the mechanism of action was hit by the Q23. And in the aerobic condition, that ATP synthesis is about 1 nanomolar activity of IC50, which is more or less equivalent to the MIC that we have seen in the bacteria. Even in the hypoxic condition, that we are seeing the similar range of activity in both in aerobic and in anaerobic conditions. So in collaboration with Novartis in Singapore, that we were looking for a target, and by generating the resistant mutant. So we have identified about 24 independent clones, and for which is resistant to Q23, It happened to be all of them have a mutation in the same spot on the QCRB gene, which is part of the cytochrome BC1 complex, complex 3 in the electron transfer chain. And this mutation is threonine 313 to the either isoleucine or alanine mutation. So this compound does have a hotspot for the resistant mutation. This is a schematic that where the Q23 works in the complex 3, and the bedaquiline is working on the, as you know, this ATP synthase. So it's in the same pathway. So we have been expecting that, you know, like there could be a synergy because ATP synthesis so far has not been, you know, like in generally known to be cannot be blocked completely by a single compound, and then if you can use the two different compounds in the same pathway, there is a lower possibility of the driving, actually the emerging of resistance, as well as the could increase the efficacy of the combination regimen. And the first, not the first, but the in vivo efficacy model was done in the established mouse model. Is 3 weeks of a pre-infection and then 4 weeks of treatment, 5 days a week, because the technician has to go home that weekend. So in the result that we tested compounds compared to the INH and INH of 25 mpk and the bedaquiline of 6.5 mpk as reported in the literature. So we do see the nice dose response of Q23 from the 0.4 mpk to the 10 mpk. Both in lung and spleen. And overall drug level that we defined the minimum efficacious dose as 2 mpk, where it showed about the 2 log reduction of the lung TB burden. And at this 2 mpk, that our assessment of the PK/PD study, this experiment has been done in the separate setting because we could not able to we could not actually get the blood out of the BSL-3 lab for the testing due to the local regulation. So the same dosing schedule has been tested in the same mouse strain, BARB-C strain, and then this is overall the C-trough, the Cmax level is 0.5 micromolar to the 1 micromolar, as to reduce the 2 log reduction in TB burden in lung. And we also tested in the same model that we also tested, this is a pilot test, I should remind you, that we also tested some combination effect with the different compounds. And first thing that we wanted to test was the synergy with the bedaquiline, because both compounds lie in the same pathway. So the Q23 and bedaquiline basically show the range of efficacy in the margin of error that we have experienced in many repeated experiments, and in the combination that actually it got reduced about 5 log reduction and showing a clear synergy. In the even more the preliminary experiments that we have surprisingly found that the Q23 and the pyrazinamide show pretty striking synergy that reducing the CFU count to the 1 log, which is the limit of detection for the CFU counting. And another peculiar feature of Q23 is in the course of the counter screening of Q23 against the human target, we have screened against about little less than 500 different human proteins, and we found that Q23 is hitting the one of the pro-inflammatory enzyme called the 5-lipoxygenase, 5-LO. And there has been a publication that says 5-LO knockout mouses have a slower progression of TB when they are infected with the TB, meaning that there is some inflammatory effect that actually helping TB infection. And we did look at the lung histopathology before and actually with the treatments with the INH and the Q23. And what you see here in the red arrows are the granulomatous that formed in the mouse model that is very abundant in the untreated model, and is still there after the INH reaching the same level of TB burden reduction. But after the Q23 treatment, you don't see this granulomatous formation. And hopefully this repeats in human, that actually that we are hypothesizing that this is the kind of efficacy that can eliminate the hiding place for the bacteria and expose the bacteria to the immune system as well as to drug. Oops. So the Q23 is currently in the phase 1 in the US, and we just finished the complete dosing phase for the single dose administration. And CSR is going to be available in a couple of months, and we will start multiple dose administration in this year. So obviously that we have a fairly good safety margin in the GLP loaded and non-loaded study, and we've been looking at the cardiotoxicity from the beginning, and this aspect has also been applied to the phase 1 design. And so far there is no sign of the QTc prolongation. And the DDI profile has been looked at, and then there is no concerning findings yet. And genotoxicity is okay. Because of this compound is hitting the electron transfer chain, and we were initially concerned about whether these compounds might be affecting the electron transfer chain of mammalian origin. And then we are testing these compounds in the mitochondria, liver mitochondria for the mouse and human, and there is no actually the inhibition of the ATP synthesis in both settings. And we also look at the oxygen consumption, which is more sensitive than the ATP synthesis, and then there is not none effect so far that up to the 161 micromolar concentration. You can find out about our phase 1 design, SAD design, from the clinicaltrials.gov. And in summary, that the Q23 is a novel anti-tuberculosis agent targeting the cytochrome BC1 complex. And so far that we found that this is active against the MDR, XDR, and TDR strains. And the good efficacy. And we just got end of last year, we got the orphan drug designation from the US FDA, and hopefully this will help a development pathway. And the picture on the right corner is our CTM. Thank you very much for your attention.
Thank you, Kiyean. Any questions for clarification?
You mean anaerobic bacteria, other bacteria?
Right, Bacteroides species, Clostridium species.
No, I think the data that I showed you on the slides are all the strains that we have tested. I don't think we have tested against anaerobic bacteria.
So I suggest you do the test on it to see what it does to the gut.
Okay, thank you. Obviously that is, this is probably too early to go for how we are going to do the later stage development. So up to the EVA study will be done in the drug sensitive patient as a standard protocol. And then we will go to the drug resistant patients in the phase 2B trial. But this is still on, at the planning stage.
Okay, Kiyean, thank you. Thank you very much.